Now we move into the discovery of the inheritance mechanism. It was Gregor Mendel who discovered a type of inheritance, Mendelian inheritance or monogenic inheritance. There are a number of other different types of inheritances, and that's the third part of this lecture series. First, let's go through a backdrop about Mendel as well as his experiments and what kind of hypothesis testing he was conducting. And eventually, he discovered the field of genetics. Entering Gregor Johann Mendel, who was Czech. He was an Augustinian monk, and he took the vow of botany, I suppose. He trained at the University of Vienna, which is still one of the most prominent schools in the world, and he had a garden, and he was working on all sorts of experiments with fertilization, cross-pollination. What he was really interested in was creating new variants, new colors of flowers often, so that when other clergymen and laypersons came by his garden, he had, an array of flower colors to display. And he was specifically looking at hybrids, and you need to hybridize to come up with new variation, and this is one way to create new color variants. And after he conducted his experiments and his notes, they were published in an obscure Czechoslovakian journal. It was rediscovered later in the 1920s and the 30s, but all the terminology he used in his paper are sometimes emphasized and used, but they form the basis, and I'll show you the differentiation between some of the ones that Mendel came up with and ones we use today, but they form the basis and foundation for the field of genetics. Scientists, botanists, and any type of field study, scientific inquiry, you usually have a model organism. In this case, that model organism for Mendel was garden peas. And so here are a few traits that he used. You may be familiar with maybe one trait, the color of the petals of the flowers or the stems or the color of the peas, but Mendel in fact had a number of different variants he used. And what he was doing was he was recording and tracking the observed variation in each one of those generations and watching how the frequencies of the phenotypes, what he was actually seeing, right? Round versus wrinkled seed shells, for example, on a pea, and looking at that variation in a population sense over time. First, let's backtrack and understand what Mendel was doing. He was testing what is known as the blending hypothesis. This is the same idea that Darwin had, very similar to pangenesis or pangenes. And what Mendel did was, if I am testing the blending hypothesis, for instance, if I take the tallest plant or in my garden or the one with the longest stems and have that cross-pollinate with the plant with the shortest stem or the shortest plant, if the blending hypothesis is correct, then the offspring should actually be an intermediary between the two phenotypes that I see, long and short. So if I measure the height of the long stem plant and measured the height of the short stem plant, and I added them together and I averaged it or divided by 2, then I would get something in the middle. And so you see this schematic here, long stem, short stem, one plus the other arrow equals something in the middle, right? For instance. So if a plant was 6 feet tall and there was another one that was 4 feet tall, their offspring would be 5 feet tall. What Mendel noticed was that his observations, even within a few generations, were able to reject the blending hypothesis. He started off with the purebred strains. We can use stem length, right? And he found that he could even predict the number and proportion of variation in each one of those generations of those crosses or hybrids. So our example here is stem length. F0 generation would include long stem plant and a short stem plant. They cross-pollinate, and the F1 generation is that first generation of crosses of the F0 or purebred strains. What Mendel noticed in the F1 is that all of the plants had long stem or were tall, right? So the offspring, none of them were intermediary. All of them looked like the phenotype of one of the plants and not the other. What he did was he had the F1 generation actually cross-pollinate. And they're all the same phenotype and genotype. And then the F2 generation actually turned out to show the short stem again, right? So if you cross-pollinate the F1, now you notice that the short stem comes back in the F2 generation. Mendel saw this and said, how did the short stem come back? First of all, the blending hypothesis is rejected based on this information. And then how did the short stem plant come back to the F2 generation? That means there has to be something masked and carried from the F1 generation to actually have that persist. And the genius of Mendel is that he then suggested that traits have to occur in pairs, and he was correct. On the basis of this information, Mendel elaborated that traits must occur in pairs. That's the simplest conclusion. And when you look at inheritance, it has to be particle based. So Hippocrates is partly right, and those particles are not one that eventually forms a blend of the two in terms of its expression that we observe, and that is incorrect and rejected and can't be used as an explanation. We call these particles genes now, of course, and each individual has 2 genes for each trait. We know this because of meiosis, right? You get 1/2 of each physical trait you have that is Mendelian in a sense from each one of your parents, right? So those gametes, they only carry 1/2 of the information, right? Because traits occur in pairs, you have homologous chromosomes, right? So Mendel was very forward thinking in his conclusion, and we were able to demonstrate that the principle or law of segregation is substantiated. So as we move into genetics, we're going to start using other terminology like genes and alleles, but we need to know what they are, right? A gene effectively is just DNA that codes for a specific protein that is a trait or a feature that we can observe, right? Or a phenotype. So you have a gene that, something that's inside your genetic code that codes for a specific protein. For instance, it's coding for your brown hair, and we observe your hair that is brown. A locus is a site on a chromosome where that gene is located. So we'll talk about some specific gene. It may have a few numbers or letters associated with it. And then we'll ask, where is the locus? And somebody will say, well, the locus is actually on the first chromosome in this spot, very specific. So these are some other terms that are going to be used that are important when we talk about different types of inheritance, as well as how genes are responsible for physical traits or phenotypes. Alleles are just an alternate form of a gene. And so allele and gene are synonymous. These terms are the same. Allele is the term that Mendel used. So oftentimes we use them interchangeably, and allele is kind of like tipping your hat to Mendel. And genes or alleles can be dominant or recessive in a Mendelian sense, of course, and in polygenic inheritance. But traits that are masked in the F1 generation, they were coded by recessive alleles. And recessive alleles are not expressed in the presence of dominant alleles. So we can think of here, dominant allele is a capital T, that's a long stem. Recessive allele is a short stem, that's lowercase t. If you are looking at Mendelian traits where there is one locus responsible for a physical trait in that site on where that gene is located, individuals can be either homozygous, right? We have 2 copies because one from each parent. You can be homozygous. Homo meaning same, zygote, that's when the egg is actually fertilized and to form you. This is two copies of the same alleles, right? So you can be homozygous dominant, meaning you have two copies of the dominant allele. So two capital T's. Or you can be homozygous recessive, two lowercase t's. Or you can be heterozygous, meaning you have one copy of each, one dominant and one recessive. This brings us to the important distinction between genotype and phenotype. I was giving you at the very end a number of genotypes like homozygous and heterozygous. These two terms, genotype and phenotype, are very important to remember when you look at genes as well as that and phenotypes. So a genotype is the actual genetic makeup. You can think of the type of genes. If we zoom in on your chromosomes on a specific locus where a gene is located, right, what is the information that is there, right? It could be the nucleic bases and nucleotides, but it also can be, well, it looks like the dominant gene that I observe, right? And so we represent that by two letters because traits occur in pairs, right? You either on that site, on that chromosome, you are either homozygous dominant on your homologous chromosomes, or you are homozygous recessive or heterozygous. Now, the phenotype is actually what you observe, right? This is what we can actually measure, right? In this case, it is the genes for tall or long and short stem. So what we're actually observing, that's a physical trait we can see, and there's a major distinction between both, right? So when you look at a plant and their phenotypes, for instance, look at a long stem plant, its phenotype. It's a long stem, it's longer than the short stem. I don't really know what its genotype is. I know it's either homozygous dominant or heterozygous because you have to have at least one dominant allele or gene to express the dominant trait. If I see a short stem plant, I know its genotype definitively because you have to have two of the recessive alleles or genes to express the recessive trait. So you can see that the phenotypes, some of them can be the same, but the genotypes different. Homozygosity is where you have the same allele. Heterozygosity, or being a heterozygote or heterozygous, is where you have one copy of each. And in fact, it's the most diverse genotype in a Mendelian sense. Let's go through step by step Mendel's experiment using what's known as a Punnett square, which is a way to demonstrate the probabilities of alleles to produce different genotypes. You have the different alleles, dominant, recessive, capital T, lowercase t. You also have your F0 generation, which includes the homozygous dominant and homozygous recessive parents, right, of the F1 generation. So this is just a graphical representation of Mendel's experiment using the long and short stem genes. So you'll notice the F0 generation, which you have both homozygous because they are purebred strains. They cross-pollinate, and you notice that there's a divide or a split between rows and columns, and the split is showing you in simulating meiosis, where you have the alleles split or segregate and go into their respective gametes or sex cells, because we know that the sex cells, because of the process of meiosis, carry half the total complement of information for that organism. So you'll notice that the F1 generation could only receive, right, each one of them, the capital T and the lowercase t, the dominant and recessive, and only be heterozygous, right? If a purebred homozygous dominant plant is contributing to the next generation of offsprings, it can only give a dominant allele, one of them. The homozygous recessive plant, the short stem, it can only give the recessive allele to its offspring. So you'll notice that if you do the probabilities on the Punnett square, all of their offspring will look the same. all of them will be heterozygous because they'll have one of the dominant allele and one of the recessive allele that they got from each parent. They're all going to look the same. They're all going to have long stems, right? Because in the presence of dominant alleles, recessive alleles are not expressed. The only way you could have a recessive plant or a short stem plant is if you had both recessive alleles. And again, this is what Mendel noticed in the first generation of offspring. Now we'll look through or go through the rest of Mendel's experiment where he took the F1 generation, had them self-pollinate. And now you have the most diverse genotype, which is heterozygous. F1 generation, all of them are heterozygous, all of them are long stem. You notice that they can contribute both either the dominant allele or recessive allele to their offspring. What this does is creates greater probabilities, and if you'll notice that the F2 generation, that you, in phenotype, you have a 75% chance that the organism or pea plant is going to be long stem. You have a 25% for it to be short stem, or 25% of the offspring are supposed to be, right, short stem because they have the recessive alleles, and that is expressed. Now notice this is what Mendel observed, that he observed that the recessive allele came back. And how did he have a recessive plant or short stem plant? It skipped a whole generation. That means that there had to be something retained. It was particle based. It was carried through a whole generation. You'll see that the genotypic ratio is a little more complex, but you'll notice that most plants looked like their offspring. But there is also a 25% chance that the plant will not and be a short stem. This is what Mendel concluded, that you have to have an inheritance mechanism that is particle based and occurs in pairs.